So we'll go ahead and get started here. Well, good afternoon and welcome to this session on basic and translational pancreatic cancer research. When I was looking at the program a week or so back, I was really excited about this session. There's some really interesting talks here that will relate some basic science and translate into clinical care of patients, and we have a wonderful lineup of speakers. It's my honor to chair this session, Chris Wolfgang from NYU, and to co-chair this session with Haomin Wang from MD Anderson, who I'm sure most of you will know. With that, I would like to introduce our first speaker, Christina Towers. Christina is from the Salk Institute. She's the associate professor at the Salk Institute. She has the Richard Hyman and Ann Daigle Endowed Chair, and she has interest in studying the unique metabolism of pancreatic cancer and searching for vulnerabilities and how that could be leveraged in the care of patients. And she'll be speaking on leveraging autophagy in pancreatic cancer. So, Christine, I'd like to hand the podium over to you.
Okay. Thank you. Thank you for promoting me right then, because I'm actually an assistant professor, not an associate professor yet. I just started my lab just a little over 3 years ago at the Salk Institute. So I'll be talking about still some work that's in progress, but work that we're doing really focusing on autophagy. I don't have anything to disclose. Okay, so I don't have to introduce this topic to this audience, of course, but pancreatic cancer is an absolutely devastating disease and on its way to being the second leading cause of cancer-related deaths. But what's interesting about pancreatic cancer is that it's quite addicted to this cellular recycling process called autophagy. And there's been a lot of work done in the field from many, and I'll highlight the work from Alec Kimmelman, as well as people like Eileen White and Rashika Pereira, who have really shown that PDAC in particular are really addicted to autophagy, and this is work here from Alec's lab showing that when you manipulate autophagy by targeting a core autophagy protein ATG4B, you can drastically reduce the growth of these tumors. So just to give a little bit of background about the process of autophagy, it's a highly conserved cellular recycling process that involves the formation of a double membrane structure called an autophagosome, which engulfs damaged cytoplasmic material, ultimately targeting it for the lysosome. So it is a cellular recycling process. We know it's important in pancreas cancer, and so there's been many efforts to target this process. Some of the most classically used drugs include hydroxychloroquine, which is FDA approved as an antimalarial, but it has poor specificity and poor bioavailability, meaning it's never going to be the ultimate way we target autophagy in pancreas cancer patients. So newer drugs are coming on the market, and preclinical work has really highlighted that some of these upstream kinases within the autophagy pathway could be viable targets, including these upstream ULK1/2 kinases. And so there are new drugs coming on the market that are in preclinical development that are much more specific and have much better bioavailability. But my lab's really interested in how can we target autophagy and what are going to be the best ideal combination therapies that we're going to need to really have an effect in our patients. So to try to understand this, what we did was we created cells that were highly resistant to different autophagy inhibitors, using both hydroxychloroquine, again, targeting the lysosome, but really also comparing that to newer drugs that target these ULK1/2 kinases, which for short I'm going to call MRT. So it's a really simple model where we just treat cells with increasing doses of these drugs until they acquire resistance in culture. This is just showing IC50 curves showing this really nice rightward shift with these drugs, so the cells are resistant. Importantly, if you treat the cells with one autophagy inhibitor, they acquire resistance to all autophagy inhibitors. So we first confirmed that they are not doing any autophagy. I won't go into all the details of these assays, but we leverage pH-sensitive and pH-resistant fluorophores like mCherry and GFP, and then we can measure autophagy quite nicely. So this is just really highlighting that as these cells acquire resistance to autophagy inhibitors, they are no longer doing autophagy. So they are adapting new mechanisms, and by understanding these mechanisms, we can identify the ideal combinations to combine with autophagy inhibition. So to start to get at what these adaptive mechanisms could be, we first just started with very basic RNA sequencing, where we compared our cells that are resistant to different autophagy inhibitors, as well as cells that have a sustained knockout of a core autophagy gene, ATG7, as sort of the ground truth. And metabolism was one of the largest sort of overarching themes that came out of the RNA sequencing. And so that really led us to investigate metabolism a little bit more. And so we partnered with people at the Salk Institute who study metabolism and metabolomics, in particular with Christian Metallo's group. And we did some stable isotope tracing. So here I'm showing a glucose trace where every single molecule within the glucose is isotopically labeled. And so we can then trace glucose as it goes through different metabolic processes. And in particular, I'm going to focus on the TCA cycle, which is in the mitochondria, and it's a critical metabolic pathway. But one of the things we noticed right away was that the key enzyme, pyruvate dehydrogenase, which is the enzyme that facilitates pyruvate's entry into the TCA cycle in the sort of clockwise fashion, that activity was downregulated as cells adapt to autophagy inhibitors. But, one more point here. This was not just true in our cells, but if you correlate autophagy signatures with pyruvate dehydrogenase activity, this was largely correlated across many different cells, so indicating that autophagy is related to pyruvate metabolism. But I just want to remind you that these cells are acquired resistance, which means they're growing, they're proliferating, they are existing in very high doses of these drugs, and they're thriving, right? So how are they doing this? Even if they have less pyruvate metabolism, what is the adaptive mechanism? And so by carefully looking through some of this metabolic tracing data, we were able to see that even though they have lower pyruvate dehydrogenase, they still had functional TCA metabolism. So even though pyruvate canonically goes through pyruvate dehydrogenase, there are alternative anaplerotic mechanisms or alternative ways for cells to get glucose molecules or to get the carbons from glucose molecules into the TCA cycle. And one of these alternative routes is through an enzyme called pyruvate carboxylase. And so this shunts the TCA cycle, so you actually go in the reverse counterclockwise metabolic pathway, still allowing carbon cycling through the TCA cycle. And so when we looked through our databases and through our RNA sequencing data, we saw that this rate-limiting enzyme, pyruvate carboxylase, was upregulated in cells that adapt to autophagy inhibitors. And the enzymatic activity of pyruvate carboxylase was also upregulated, and we can see that by the ratio of the carbons that are coming into the TCA cycle through oxaloacetate or aspartate versus the full pyruvate. Okay, so a little bit about, why is PDH activity downregulated? So a recent paper had come out showing actually that nucleotides, and in particular pyrimidines, are upstream of pyruvate dehydrogenase activity, and this is mediated through a rate-limiting enzyme of TPK1. So TPK1 was downregulated in our cells as they acquire resistance to autophagy inhibitors, which really led us to investigate nucleotide metabolism more broadly. And so, in fact, nucleotide metabolism, and especially pyrimidine metabolism, was affected in our cells that are acquiring resistance to these different autophagy inhibitors. And when we look at nucleotides broadly in our cells, we saw that they were really depleted in these cells that were sustaining growth without doing autophagy. Okay, so I want to pause for just a quick second and give a little bit of background on nucleotide metabolism, and in particular, pyrimidines. How do cells maintain their pyrimidine pools? Well, there's 3 ways that cells can do this. Eileen White has shown that autophagy can help maintain pyrimidine pools because it breaks down nucleic acids to sustain these pyrimidine pools. But of course, cells can also de novo synthesize their pyrimidines, and this is through a rate-limiting enzyme of DHODH, which resides in the mitochondria. And finally, cells can salvage pyrimidines from the extracellular space. And so in a tumor, this would be from the tumor microenvironment. So if our cells can't do autophagy, and yet they're growing, they're dividing, which means they must be synthesizing RNA, they must be synthesizing DNA, how are they maintaining their pyrimidines? And so to investigate this, we looked at some of these rate-limiting enzymes, including DHODH and CAD, which are critical for the de novo synthesis pathway, as well as the enzymes that are critical for the salvage pathway. And the de novo pathway, what you can see, is actually downregulated, whereas many of the enzymes and the transporters in the salvage pathway were upregulated. So they're not doing the de novo mechanism, but instead they're salvaging their pyrimidines from the extracellular space. And we hypothesized that we might be able to leverage this because, as we know, many of the standard of care drugs that are used to treat pancreas cancer look like pyrimidines. And so if cells are salvaging pyrimidines, would they salvage pyrimidine analogs? And would they become more sensitive to this? So we treated our cells that are resistant to different autophagy inhibitors with a panel of different drugs, both standard of care drugs as well as autophagy inhibitors. And what we saw was actually mostly pan-resistant. So anything above the line here is a rightward shift in an IC50 curve, and anything below the line is a leftward shift in the IC50 curve, indicating sensitivity. The only drugs that these cells were sensitive to were pyrimidine analogs. And interestingly, I'll point out 5-FU, of which they are not sensitive to, but 5-FU is actually the only pyrimidine analog which is metabolized through the de novo pathway. All of the others are salvaged similar to pyrimidines themselves. Okay, so just to emphasize this point another way, this is live cell imaging where you can see the blue lines are cells that are resistant to autophagy inhibitors. The purple line there is treating those cells with gemcitabine, which is a pyrimidine analog, and you can see that it completely wipes these cells out. They are extremely sensitive to pyrimidine analogs. We showed mechanistically that this is in fact due to the fact that they are salvaging more. So if we knock out any of the enzymes that are critical for pyrimidine salvage, we can completely rescue this sensitivity to gemcitabine. This is true in vivo as well. If we orthotopically transplant these cells into mice, we can see that using a very low dose of gemcitabine, which has no effect in the control tumors, can drastically shrink these tumors that have acquired resistance to autophagy inhibitors. So this is all nice in an acquired resistance setting, but we were also really interested if this would mean, if this would indicate that we could have combination effects with autophagy inhibitors and pyrimidine analogs in patients who have never seen an autophagy inhibitor. So to do this, we wanted to use drugs that were more bioavailable and better amicable to move forward in the clinic. And so we partnered with a chemist here on the Mesa at SBP, as well as with another lab at the Salk Institute that's led by Ruben Shaw. And they had a long-standing collaboration to understand more about these ULK kinases, these upstream kinases that regulate the autophagy pathway. And in doing so, they had identified that within this complex, there's the ULK1/2 kinases, but there's also these additional autophagy proteins, in particular ATG13, which is a core autophagy protein. And they found that actually the best targeting drugs that could hit these kinases were the ones that led to ATG13 degradation. And so here's just a quick assay showing that ATG13 gets degraded when ULK1 or ULK2 is hit in its kinase domain. And by correlating the best small molecules that could cause ATG13 degradation, they were able to identify novel ULK1/2 kinase inhibitors. And so in using these new drugs, and in particular, we're using one called SBP1750, we were able to show that these drugs do not cause any toxicity when combined with gemcitabine in mice. However, they do significantly reduce the growth of these pancreas tumors when they're orthotopically transplanted. Importantly, we can confirm on-target activity of this combination. And I want to pause just to say that one of the biggest challenges in the autophagy field is that we do not have good biomarkers for efficacy with any of our drugs. This is the first time where a drug can we have a biomarker and we can use the degradation of ATG13 to confirm that we have on-target hits. And so in the tumors, we can really nicely see that ATG13 is degraded, indicating that we have a good drug with bioavailability that is actually hitting autophagy and has a combinatory effect with pyrimidine analogs, of which are standard of care for pancreas cancer. Okay, and so what I just summarized today, and this work is now published on bioRxiv as we're in the process of publishing it in peer-reviewed journals. And so I only had time to really hit on the highlights, but what I've shown is that when you target autophagy, as many cancer cells will die from this because they require autophagy to survive. But as these cells adapt, they can sustain new mechanisms to survive. And so what we've shown is that in these pancreas cancer cells, you have multiple ways of maintaining pyrimidines. Autophagy is one of them, but they can also de novo synthesize or they can salvage their pyrimidines. But when we block autophagy, what ends up happening is that the de novo pathway is not active, and I didn't have time to go into this, but this is because the mitochondria themselves are less active, and so as they have less de novo metabolism, and this is also because the pyruvate metabolism is altered as well, which I did touch on a little bit. But because they cannot de novo synthesize their pyrimidines, it causes them it causes them to, as they're adapting, because they cannot de novo synthesize, it causes them to salvage instead. So they upregulate pyruvate metabolism, which I talked about a little bit here, as they attempt to do de novo synthesis, but because they don't have the rate-limiting enzymes, they can't. And so instead, they salvage through the salvage pathway. But we can leverage this adaptive mechanism to treat pancreas cancer tumors. Because these are standard of care drugs, we can combine autophagy inhibitors, and they will become hypersensitive to these pyrimidine analogs. And so by really understanding these mechanistic and metabolic adaptations, we can leverage these to come up with ideal combinations. And of course, today I talked about combining them, but really what I think what our work highlights is that if we can pre-treat with autophagy inhibitors, we can highly sensitize our tumors to pyrimidine analogs. And so the next steps are to really understand sequencing of these different drugs. And if we could first cause this adaptive mechanism to take place, that might allow us to use lower doses of pyrimidine analogs and increase sensitivity to these drugs. We're also really trying to understand what cells within the tumor microenvironment are providing these pyrimidines for the cells to salvage. And so could targeting the tumor microenvironment provide an even additional synergistic effect? Okay, and so with that, I will stop and thank the people who did the work. It's really been a whole lab effort, but I'll highlight Suzanne Dufras, who is a very talented postdoc in the lab, who's really led this project. And I really want to thank our collaborators at the Salk, especially Christian Metallo, as well as Ruben Shaw and Nick Cosford's lab on the Mesa. And I guess I won't take questions, but I will take questions at the end. Thank you.
Yes, thank you very much for the wonderful lecture. So we will hold all the questions to the end of this section. So the next speaker is Dr. Hannah Orgel from Germany. His lab is mainly focused on pancreatic cancer metabolism, inflammation, and immunoregulation. So today he will talk about his title is Pancreatic Cancer Immunoregulation. So this talk will be virtual, so they'll play the video for you. Thank you.
Ladies and gentlemen, dear friends, thank you very much for having me virtually to the APA meeting in San Diego, to which I was unfortunately not able to be there in person, but I'm very grateful and happy that I was given the opportunity to share my thoughts on how the immune system is regulating pancreatic cancer and whether the immune system is a potential target for the treatment of pancreatic cancer. Today I would like to focus on the NF-kappa-B REL signaling pathway to demonstrate to you how we are trying to understand in more details how pancreatic cancer can be made more sensitive to immune therapy in the future. Whenever I talk about pancreatic cancer and immune system, I start with a case that really struck me and that happened in our hospital. That was a 53-year-old male patient who presented to us with a diagnosis of a metastasized pancreatic cancer 5 years ago, and this patient, quite young patient, was treated in a different hospital with all the classical therapeutic regimens, starting with a clinical trial in which he was enrolled, gemcitabine plus paclitaxel and other drugs combination, and then he received FOLFIRINOX after progress, and even after FOLFIRINOX, this patient was sent to a hospital in which they performed TACE to treat the liver metastasis, which was very predominant, as you can see here on the slide, huge liver metastasis. This patient presented then to our outpatient clinic, and I saw this patient. I performed a genetic analysis in this very young patient who was in a very good health condition, and the genetic analysis came back with a high PD-L1 status, classical KRAS G12D mutation, P53, and also an FGFR amplification. We put this patient on a combination that has not been given so far to any patient, which is pembrolizumab and futibatinib because of the amplification of the fibroblast growth factor receptor and also because of its high TPS score. And then we followed up this patient, and until last month, this patient showed a durable response to this combo therapy of pembrolizumab and futibatinib. And as you can see with the tumor marker, that the patient rapidly responded to this combination therapy and the liver metastasis started to shrink, and the recent analysis that was performed in October clearly showed that there is basically no liver metastasis anymore in this patient, and the primary tumor, which was initially localized in the pancreatic head, is not visible anymore. And this is a very good case that teaches me, at least, that the clinical trials that were done with immunotherapy in patients with deficient MMR, like microsatellite unstable tumors, that were negative basically for two tumor types, for pancreatic cancer and for glioblastoma. That clinical trial was really disappointing for all GI oncologists, showing that pancreatic cancer is even resistant to these very efficient immunotherapy. So, but this case taught us, okay, there is much more behind that, looking for a deficient MMR and deciding whether a patient is eligible for immunotherapy. We need to understand in more detail why pancreatic cancer is so resistant to immunotherapy. And this is due to the fact that tumors are defined as cold and hot tumor, and that hot tumor are eligible for immunotherapy. But what exactly defines the fate of a tumor, whether it is a cold or a hot tumor, is not really and clearly understood. But we consider, together with other scientists, that NF-kappa B signaling might be a relevant mechanism to define the fate of a tumor, due to the fact that NF-kappa B is involved in many mechanisms that are so relevant for a hot tumor, such as inflammation, microenvironment, angiogenesis, and immune response. And this is why we started to focus our interest on the NF-kappa B signaling pathway years ago to understand its contribution to pancreatic cancer. In the past years, we tried to understand the contribution of the single subunits that are very relevant to the pathway of NF-kappa B to pancreatic cancer. We studied the role of REL-A during pancreatic carcinogenesis and found out that the NF-kappa B subunit REL-A is critical during pancreatic carcinogenesis through its contribution to the senescence-associated secretory phenotype, and that the abrogation of NF-kappa B REL-A resulted in an acceleration of preneoplastic lesion development to pancreatic cancer due to the fact that senescence was not possible in the preneoplastic cells anymore. REL-B was shown to be relevant for metabolism in the tumor cells, but not really relevant for immune response in pancreatic cancer development. The role of CRL in pancreatic cancer has been unstudied so far. So this is why we focused on studying the contribution of CRL to pancreatic cancer development and its potential role in the immune response of the tumor. In fact, past years and several publications centered around the role of CRL have confirmed that the activation of CRL in immune cells is very important for the responsiveness of tumors towards immune checkpoint inhibitors, as shown with melanoma in the group by the group of Sankar Ghosh, and other studies have also confirmed that CRL is important in myeloid-derived suppressor cells for cancer immunotherapy, and also the activation of CRL seems to be important in fibrosis, a parameter and a mechanism that is also very important for the formation of the tumor microenvironment, particularly in pancreatic cancer. So for us, there was evidence that CRL is an important regulator of immune response in different tumor types, but has not been studied in pancreatic cancer. This is why I entrusted Derya Bakirdogan to understand the role of CRL in pancreatic cancer. She started with staining of different human pancreatic cancers to see whether this protein is expressed and where it is expressed. And she could see in 3 different human pancreatic cancer that the expression of CRL is there, and there is also a localization in the cytoplasm as well as the nuclei. And if we look whether CRL expression has anything to do with overall survival in pancreatic cancer, we performed this analysis to see that tumors with a low CRL status or expression do have a favorable outcome in comparison to those tumors with a high CRL expression. If we categorize the tumors according to the cytoplasmic and nuclear expression of CRL, and we know that CRL exerts as a transcription factor its role in the nuclei, we could see that particularly those with a nucleic expression of CRL have a bad outcome compared to those with the cytoplasmic expression of CRL, suggesting that CRL in the nucleus is somehow affecting the outcome of patients with pancreatic cancer. The expression of CRL is also confirmed in human pancreatic cancer cell lines in different types, but also in the murine cancer cells. And if we look at the tumor tissue of the pancreatic cancer model CK and CKP, we could see that the expression of CRL is detectable in the primary tumor as well as in lung and liver metastasis. And to show that CRL is activated in pancreatic cancer cells, we performed this immunofluorescence analysis and stimulated the cells with TNF-alpha and put leptomycin B to inhibit the nuclear export of CRL, and we could see that upon TNF-alpha stimulation there is a nuclear localization of CRL which is not detectable in not treated cells. To understand the in vivo role of CRL in pancreatic cancer, we generated 2 mouse lines. One which lacks CRL in the pancreas, which is called the REL knockout, CCKP, and also a gain of function mouse line in which CRL is overexpressed specifically in the pancreas. We compared these 2 mouse lines to the CKP mouse line, which harbors the KRAS G12D mutation and lacks P53 on both alleles. Looking at the survival curve, we could see that overexpression of CRL, mice overexpressing CRL, has a worse survival outcome in comparison to those mouse lines which lack CRL specifically in the cancer cells. Looking at the morphology of the tumor, we could see that CRL lacking pancreatic cancers are in most of the cases tubular cancers, some of them solid and cribriform cancers. If we compare this morphology to the CRL overexpressing mouse tumors, we could see that the solid tumor are more predominant in comparison to the other mouse lines. So this is why we performed numerous experiments to understand the contribution of CRL to pancreatic cancer development. On the one hand, we have seen that with regard to CRL expression, the outcome is worse with high CRL expression, and in mice with serial knockout, specifically in the pancreas, the outcome survival was better. On the other hand, we also observed that the phenotype of the tumor is different, particularly in those tumors with high expression of serial. The tumors in these mice were more solid with a strong environment desmoplastic reaction. So this is why we wanted to understand how serial contributes to this phenotype. Interestingly, we found out that serial overexpressing cancer cells demonstrated a greater colony formation of colony forming frequency in comparison to the other mouse lines. This was quantified in matrigel, and also in spheroids, in two-dimensional spheroids under adherence-free conditions. We could see that serial overexpressing cells showed a larger formation in comparison to the other mouse lines, also quantified here. And interestingly, we tested the surface expression of predefined stemness-associated markers, including EpCAM, CXCR4, CD44, CD133, SCA1, and also CD61, which is integrin beta 3. We tested this in adherent and in non-adherent serum-free spheroid cultures, and to our surprise, all the markers, either individually or in combination with EpCAM, were reduced in serial overexpressing cells, except for CD61 in spheroid culture. So what is the contribution of serial to organoid formation, spheroid formation, and CD61 expression? Interestingly, we found out that CD61 is also detectable in the tissue of serial overexpressing tumors. As shown here with the immunofluorescence analysis, we could detect numerous cancer cells that express CD61 in the stroma of serial overexpressing cells, which was not the case in the other mouse lines. This has been quantified and also confirmed with the graph over here. At the same time, fibronectin positive cancer cells were also higher detectable in serial overexpressing cells, suggesting that these tumor cells that are isolated in the stroma of serial overexpressing tumors are somehow under isolation stress. They are detached from the tumor formation, are in the stroma, and to survive, they seem to have overexpressed CD61 and are attached to fibronectin, one suggesting that serial in the tumor cells is contributing to isolation stress survival of tumor cells. To assess the direct interaction of serial with these genes of interest, CD61, which is integrin beta 3, and fibronectin, we performed cut-and-run experiments using the Degron system. The Degron system is a very suitable system to understand the interaction of transcription factors with the DNA. It allows the on-demand selective and reversible protein degradation, and in this system, proteins are targeted to allow micrococcal nucleus recruitment and DNA cleavage, releasing target protein DNA complexes. So to accumulate serial in the nuclei, we co-incubated the cells that we established for the Degron system with leptomycin, so that we can have the concentration of serial in the nucleus, not allowing them to leave the nuclei. And we performed the cut-and-run experiment, which is a good alternative to chromatin IPs, and then analyzed the interaction of serial with the regions of interest. And we could in general see that CREL is interfering with numerous intragenic, but also with promoter regions. Analyzing fibronectin and integrin beta 3, we could see that CREL is indeed binding to the promoter of fibronectin as well as integrin beta 3, and that it is transcriptionally active. And if we co-incubate the cells with indole acetic acid, which induces the CREL degradation on demand and selective, we could see that the peaks in the RNA-seq analysis disappear, which is true not only for fibronectin but also for integrin beta 3, suggesting that CREL is indeed interfering with the promoter regions of these two genes of interest. And also we analyzed CREL mRNA levels and with using the TCGA data sets to see whether there is a correlation between CREL expression and the mRNA and the expression of fibronectin and integrin beta 3, and in both cases we could see that there is a correlation between CREL expression levels and fibronectin as well as integrin beta 3. Given the positive effects, regulatory effects of CREL on the fibronectin 1 beta integrin 3 axis, both in vivo and in vitro, we hypothesized that the absence of fibronectin, which attaches to CD61, contributing to the survival under isolation stress conditions, that the absence of fibronectin 1 in cancer cells might counteract the CREL-induced phenotypes. This is why we generated another mouse lines which lacks fibronectin 1 in CREL overexpressing tumors. Looking at the survival, also the morphology type, we couldn't see any big difference between CREL overexpressing tumors and those with CREL overexpressing tumors and fibronectin 1 deletion. So there was no rescue in the primary tumor with the additional deletion of fibronectin in those cells that overexpress CREL in the cancer cells. But interestingly, fibronectin seems to be necessary for the CREL-induced isolation stress resistance in cancer cells, looking at the organoids and spheroids formation. And I want to remind you of the slides that I showed you before, that CREL overexpressing resulted in big spheroid formation, organoids as well as spheroid 2D formations. And this phenotype shown here is completely rescued upon deletion of fibronectin. So suggesting that in fact that fibronectin 1 is responsible with interaction with CD61 or beta integrin for the spheroid formation and for the survival under isolation stress conditions. So our results indicate the involvement of CREL in cellular fitness, so to say, during isolation stress under adherence-free conditions, which is an important concept in the metastatic cascade. And to assess the importance of the CREL-mediated integrin fibronectin signaling axis in the metastatic cascade, we utilized IV injection of cancer cells followed by lung histopathology analysis. And as expected, both the relative metastatic colony area in the lungs and the relative lung weights shown here were increased in the CREL overexpressing cells. However, upon deletion of fibronectin, the metastatic capacity is strongly reduced, suggesting that fibronectin orchestrates with CD61 to contribute to cell survival under isolation stress. This sketch shown here brings together what we have analyzed in pancreatic cancer with respect to CRL. We have shown that CRL in the tumor cells is contributing to survival under isolation stress conditions through the upregulation of isolation stress response, to the upregulation of isolation stress response with adaptation to isolation stress through upregulation of CD61, which is interacting with fibronectin to confer to the tumor cells that overexpress CRL a stronger metastatic capacity and survival under these conditions. So Daria has functionally analyzed the contribution of CRL to the cancer cells. So this is why we can confirm through our analysis that CRL in tumor cells is also contributing significantly to the biology of the cancer with respect to metastasis and survival under isolation stress, which is one important factor in metastasis. But interestingly, and coming back to my initial idea, how to overcome immune therapy resistancy of pancreatic cancer, we also tried in preliminary data to analyze whether targeting CRL, now
So, sorry about that. I think there was some miscommunication in length of talk. So it was a very interesting talk. We'll cut it there. We'll play the remainder at the end if we have any time, but we definitely want to leave time for discussion. And we have some very interesting talks coming up, but I apologize for that. So it's my pleasure to introduce our next speaker, Ziling Shen, who is professor of GI Medical Oncology at MD Anderson. He has had a career interest in studying what I think is really novel, and it's the brain or neural gut brain tumor stem cell axis regulation of tumor growth, tumorigenesis, and physiology. And he's going to be giving a very interesting talk on the vagal modulation of the brain-liver axis to alleviate cancer-associated cachexia. So with that, I'd like to turn it over to Dr. Shen.
Thank you very much. And this is my first time coming to the APA meeting. What a wonderful meeting. And it seems I've been working on the wrong organ the whole life, and it's not too late right now. With that, I'll just advance to my talk today about really kind of thinking about new ways to treating cachexia. Again, thanks for the yesterday's evening session, especially Chris, great introduction about cachexia. I don't really need to, oh, by the way, yeah, this is the companies I formed. One of the company actually I'm going to talk about the data. Actually, we formed the company and I'm going to sponsor the trial. But anyway, for cachexia, needless to say, right, many people suffer from that. And it's a very paradoxical disease there where the patients are losing weight, but they're losing appetite. The muscles breaking down, but the urea cycle shuts down. So the body's doing, and the fat turning from white to beige. So the body's doing everything the opposite of what we should be doing, as kind of Chris summarized yesterday. And besides directly killing patients, Yeah, so it's advancing here. I guess a two separate version.
We're seeing it here, so it just needs to go up on that.
Great.
Sorry about that.
All right, no problem. So right, and also when patient had a severe cachexia, then they had to stop treatment and often gets relegated to hospice, deprive them of any chance of fighting the tumor. And for this community, of course, pancreatic cancer has a very high, the highest incidence of cachexia rate, estimated to be about 80%. And there has been, right, not lack of trying. We have been hitting on all these different mechanisms, and they all look great, but then kind of ultimately didn't quite pan out in the clinic. But there's kind of a new energy getting instilled into the field, right, as Pfizer announced their anti-GDF15 enter into phase 3, and that kind of really be a boost. And it's, even I talking to, right, investors community, people never heard of the term cachexia, and suddenly everyone's saying, oh, that's actually a disease you can treat versus this a terminal condition that don't waste your time on it. But of course, if you look at their phase 2 data, they're still, right, it has a great weight gain benefit, but still there are a couple limitations. There's no survival benefit. Of course, they are late-stage patients, and also it's not clear whether patients don't have high GDF-15 will benefit from this treatment, and we'll see from their phase 3. Now, so when I thought about this disease, when I first heard about it, so I was also in charge of the Chan Zuckerberg gut-brain axis and also the DARPA electrics. I was trained as an electrical engineer and the electrics program. And what kind of hits me is there are all these different cancer types, different genetic drivers, but kind of driving towards a very similar phenotype all across these patients. And the body's doing exactly the opposite of what it should be doing, suggesting there might be a central axis that's kind of going haywire, right, in these patients. And one thing jump out is the vagus nerve. For people who are not very familiar with the neurobiology, the vagus nerve is part of the parasympathetic nerve system. And the function is, if you think about ancestor, there's a facing a lion, you have the fight or flight response. And after that, the vagus nerve kick up to quiet down the body and shuts down a lot of the metabolic processes. Similar, if a person's drowning, then the vagus nerve is going to come up and slow down the body. So now you can imagine if that gets triggered in the body in a chronic condition, and the vagus nerve actually innervates all these organs that are related to cachexia, goes up afferent onto the brain and downstream to all these metabolic organs. So my very talented back then, the student Alicia, now she's became assistant professor, did this vagal recording in the animals. On the top is the normal animals. You can see that's a typical vagal recording. On the bottom, these it become highly activated, right? The vagus nerve become very noisy. And so you can see a hyperactivated, and this is the quantification of the rooming squares and the peak values. You can see how different the vagus nerve is acting. And that would have implication on all these metabolic processes in all these organs, as I just talked about, because the vagus regulate the appetite, the food choice go down, liver metabolism, right, the gut, and the pancreas. So again, but is that really true in patients? So we actually look at some patients, we look at 29 patients, pancreatic patients, and really the level of acetylcholine, which is a vagal neurotransmitter in the peripheral system, is highly upregulated in the cachectic patient. And furthermore, that started before the cachexia actually developed into a severe form. The progression, it actually correlates the progression to the cachexia. So then we use the standard cachectic model. The LLC is the lung cancer model. The orthotopic KPC is a pancreatic model. For the rest of the talk, I'll just focus on the pancreatic data, but everything was repeated in the lung cachexia model too. And here we perform a vagotomy, which is cut off the vagus nerve on the cervical side, and which has used to be FDA approved as a procedure. So you can see that here, indeed, when we did a vagotomy, we actually greatly restored the weight and also prolonged the survival of those animals. And furthermore, it also enhanced across the board their social activity measured by open field and and chamber, and also their muscle strength, their muscle area, right? Also by grip test, the muscle atrophy gene expression level, was kind of restored, and furthermore, the appetite improved significantly in these animals. And furthermore, also they synergize with chemotherapy because now, again, we wait till 5% loss, weight loss, which is the definition of cachexia. Then when we applied the vagotomy, the weight, not just the weight came back, but we plus the chemotherapy. If vagotomy alone achieve about similar benefit as the chemotherapy, but now you can give them chemotherapy a lot longer. So actually for this animal, when we did a vagotomy and a chemotherapy, often they don't die from cachexia. They end up having to be euthanized, often because the tumor burden become too big, so they become immobile, or the skin kind of lesion bursts open because the tumor is too big, or the KPCs, it just kind of spread all over the organs, right? So, and furthermore, but I want to emphasize the benefit came from vagus is really prolonged, right? A prevention of cachexia. Vagotomy alone does not reduce the tumor burden. Only with chemotherapy, then you further reduce the chemo burden. So what's causing this weird behavior in the cachexic, animals? And here I won't go into details. The paper actually is published. But what we found out is actually what happened in the cachexic patient, the inflammatory cytokines such as CCL2 is driving this neural information in the vagal dorsal motor nucleus. And where really kind of the vagus nerve become, the cell body become inflamed and driving this hyperinflammation phenotype. And again, right, there's also a recent paper in the Tabaz's group showing it also goes to the brain and affects these brain neurons. So again, right, this really, you can see the infiltration of this macrophage-driven, right, in these caucasic animals, shown by these different markers. And furthermore, the CCL2 level, as I said, was highly upregulated in the cerebral spinal fluid as well as in the blood. And furthermore, that also correlates in the same patient group. You can see CCL level upregulation in the blood and also correlated with the progression. Now, if we block CCL2 using either antibody or knockout animal, here I just show kind of one set of experiment. It restores all these different, the appetite, the functions, and the weight, but not to the degree of vagotomy. And furthermore, if you look at the survival, you can see a trend, but it's not statistically significant, suggesting that, right, the CCL2 doesn't completely abrogate the vagal behavior, which makes sense because if you really look at this patient and also animal models, CCL2 is not the only cytokine, right? There's redundant mechanism, and that's also show that why clinically, right, often these targeting a specific cytokine often don't achieve the effect in the patient, that there's just a lot of redundancies, but they all had to go through this vagal central axis to activate this paradoxical response. So, how does the vagal dysfunction causing the caucasic phenotype, right? So, as I said, actually in human, very recent studies show that left and vagus, right vagus actually does different things, slightly different things. Our left vagus control like the motility, heart rate, and the respiration. Or right vagus upstream actually regulates the appetite, food memory, food choice. Go down actually regulates a lot of metabolic functions. So, anyway, so in this case, we actually targeted, we showed that here, not showing here, we showed targeting the right vagus is much more effective than left vagus, which makes sense. But second is we want to ask question, right? Does the benefit mainly came from afferent, means we are changing the appetite, the food, the food choice, or downstream, basically the metabolic functions. So you can see here, both of afferent and efferent has an effect. However, blocking efferent has a much stronger effect than afferent, showing that it's not just the appetite, but also the downstream metabolic function, which actually makes sense because if you're thinking about you can't cure you can't treat caucasia by just force feeding, right, nutrients. And so when a downstream, the vagus actually innervates the liver. Now, when we notice that in these caucasic animals, the master regulator in the liver, HNF4 alpha, has been severely downregulated. And furthermore, if you culture these hepatocytes and you put acetylcholine, the vagal neurotransmitter on them, actually this downregulates HNF4 alpha. And furthermore, if we knock out HNF4 alpha, it actually replicates all the caucasic phenotypes, right? Weight change and food. So suggesting that there's one mechanism is the hyperactivated vagus nerve dumping all this acetylcholine in the liver or in the peripheral organ in general, as I show later, and shuts down HNF4 alpha and shuts down liver metabolism. And if you do vagotomy, you can see that right, downregulation of HNF4 alpha is mitigated significantly. So if we perform vagotomy, what you see is, look, we restore HNF4 alpha, restore all this liver function, restore this liver metabolism, metabolites, including even short-chain BCAAs, branching amino acids, really can restore a lot of these metabolic functions, and also things like a urea cycle, which you need if your muscle breaking down, you want to recycle those amino acids. So this actually just was published last month, so I won't go to too much detail, but overall what we found out is this systematic loop where inflamed cancer is driving systematic inflammation, especially macrophages, into the hindbrain, including DVM, including the nodules, and that causes a hyperactivation of the vagus nerve, then upstream affecting appetites and food choice, and downstream affecting the liver metabolism, and shut it down. And furthermore, again, working with Dr. Teresa Zimmers, and many of you probably know, right, one leader in the caucasic field. This is just a partial story, right? And also the liver here, we didn't talk about in the paper, is in the liver, what it really does is two things. One, you having this hyperactivation vagus nerve, besides shutting down through the HNF4 alpha, besides shutting down the metabolism, it's also activating the acute phase genes, then causing this, acute inflammation that forming a perpetual loop, right? Things like lipocalin-2 really in the liver really gets upregulated by this mechanism. Okay, so that's actually great. How can we do that to treat cachexia, right? As a basic researcher, first I was very, naive, like, oh, let's just do vagotomy on the patients. Clinicians, no way to do vagotomy on the cancer patients. So we had to find a way to block that vagus nerve without this invasive surgery. So, and this has never been shown. You can actually block a vagus nerve transcutaneously without an invasive surgery. But here we actually kind of change the thinking a little bit, right? All the neuromodulation have been targeting the sodium channel, which is fast acting, take a lot of power. But we actually switch a strategy targeting the potassium channel, which is slow acting channel. You need a very low frequency wavelength. You deplete the potassium from the cell neurons, and it takes about 30, 40 hours for the cell to trafficking all the potassium in to refire. So with that, with a low frequency signal, suddenly actually we hypothesized we might be able to transcutaneously block vagus for the first time. We did this little non-invasive patch, and indeed it showed it's very effective blocking the vagus nerve. And also the effect actually lasts right, more than 30 hours. You can see the acetylcholine, the green line goes down, and took about 38, more than 30 hours, almost 40 to come back. Similar, and you can, that's a vagal recording, and this is the acetylcholine in the blood, right? A little delay, but kind of follow the same trend. If you look at channel 4 alpha, there's, it doesn't come all the way up. You need like a daily dose, right, to bring it up, but it's all kind of consistent. And furthermore, also for this audience, I just want to say it's not just the liver, right? When you're writing a paper, you have to keep it simple, but you can see, right, the acetylcholine level in the stomach and pancreas both are going over the roof for these animals, and when you perform this blocking, you actually also block that. So that's why it's so effective. It's really targeting systemically, not a one single mechanism. And furthermore, to restore the weight, having the, the synergy with the chemotherapy, restore all those, functional parameters as I just mentioned. Okay, so would that really work in human, right? We're gonna try the translation. So then we test, so the question is the mouse, the vagus nerve is much closer. In human, would that work? We actually test the harder model, which is pig. For pig, there's about 4 centimeter fat between the vagus nerve and the skin. For humans, about 1 centimeter fat. So if it works in pig, it's probably going to work in human. And here we really kind of did this kind of a wearable block and test it. So you can see initially it's at the basal activation, and we put an upstream stimulator that mimic the hyperactivation of the vagus nerve. Turn on the blocker, shuts it down, right, very cleanly, 30 minutes. And after that, you stop blocking and go back to the basal level because it doesn't have enough potassium to do that hyper stimulation. And we also show that, right, the vagal block is indeed very safe. It has no effect on heart rate and respiration, all that stuff, and also no EKG, no arrhythmia. And furthermore, there's no irritation to the skin and also different organs, and no toxicity. So after that, again, by putting to the patients, there's also very important about compliance, right? The patient gonna take the device home. How do you know the patient put it on correctly? How do you know the patient's turned it on even? So here, right, we have to develop this whole device that we are gonna be measuring impedance to ensure that the patient put on correctly. It's recording the on and off time, so really allow us to track the compliance. And also test on me. Actually, the true story is my scientists actually test on me before test on the pig, because PI is very easy to replace. The pigs are much more precious. Took her a lot more effort to get the pigs in. So she's like, I want to absolutely make sure it works before I put on pigs, and you look, safe enough. So anyway, so yeah, it's a wearable sensors, 30 minutes daily, 40 hours, placed on the right side of vagus nerve. It's a flexible electrodes, which is actually replaceable, and also it's using low frequency, and it's rechargeable. So, but ultimate question, would that work in human, right? But how are we gonna test that, get a first human in human data before I should put on the cachexia patients? So here actually, my dear colleague, Dr. Scott Kopet, who is my co-director of the colon cancer moonshot at MD Anderson, kind of bring up this very fascinating thing, like you probably met doctors here are learning their first year of medical school, which is this kind of Valsalva maneuver, right? If you blow into a narrow hole, and more strongly, if you're in a fetal position, you are mimicking a drowning experience, and your vagus nerve is going to jump up. So you can see that, right? That's kind of what you do. If you do a Valsalva nerve, put a device on, complete block the heart rate. How long that effect going to last? In animals, 30, 40 hours is true in human. We did longitudinal Valsalva. You can see initially, right, you get a heart rate drop. As soon as the device on for 30 minutes, that effect lasts for 30 hours until that come back out. So again, so now the trial will be starting in the end of January at MD Anderson at GI Medical Oncology with 20 patient leading trial that we actually going to monitor patient daily to make sure the device is working, and with a blood surrogate marker followed by 120 randomized trial for pancreatic cancer patients. So anyway, here I just want to concluding, right, that here we actually discover a interesting previous undiscovered gut-brain axis that potentially using a transcutaneous device, we can block that device and treating the patients. Here I want to thank kind of the team, but really Alicia is the unsung hero. She'd worked on this for 4 or 5 years when we had an idea just by herself. No one believed it, and kind of really made the whole thing work. Then we kind of, Brandon will be, Dr. Smaglo is leading our clinical trial in January, and with our collaborator, Dr. Russ, and also Narma at the Weizmann Institute, who kind of bring that liver angle into our mechanistic understanding. Thank you very much.
Thank you, that's fascinating. Our last speaker for this section is Dr. Lei Zheng from University of Texas, San Antonio. To many of us, Dr. Zheng is a very well-known, very distinguished researcher. Today he will speak to us on the why pancreatic cancer with lung metastasis versus liver metastasis have different outcome, the biological and the clinical reasons. Thank you.
Thank you for the introduction. So I'm actually a translational researcher, so I always actually ask questions clinically. And so the story is like that. So for a medical oncologist, they think actually either do surgery. If you cannot do surgery, you chemotherapy. If a metastasis, chemotherapy. And unfortunately, there's a very limited option of the treatment for the metastatic cancer. So this is we listed the treatment for the metastatic pancreatic cancer. This is a review actually written together by me and Chris Wolfgang. And so this is really cheating. We put everything actually related to the pancreatic cancer here. But as you can see, some of the treatments, the target therapies, is for the genetic alterations only occur maybe at 1 or less than 1 out of 1,000 patients. So essentially, our treatment for pancreatic cancer is still limited by the 2 lines of the chemotherapy. One is called FOLFIRINOX, the other is gemcitabine and Abraxane, or there's a alternative form for the FOLFIRINOX is NALIRIFOX. And so that's why when I started my career in pancreatic cancer as a medical oncology, I looked for some other option beyond the medical oncology treatment. So I steal a cohort from Chris Wolfgang. It's about only 200 patients on the left. I did this analysis. This is a patient that had a surgery at Johns Hopkins and look at their recurrence pattern. My idea is just to see actually whether all the metastasis recurrence are the same. And so I have the interesting finding, which is very consistent with our clinical observation. At that time, no one actually has done the summary. So we find that the patient who after surgery had a lung metastasis as the only site of the recurrence, even the lung metastasis as the first site of recurrence, followed by the recurrence in other site, the patient has a much better outcome than even for those patients with a local recurrence. So from that study, we think the patient with a local recurrence probably is not truly the local recurrence, actually. It just happened to be the systemic recurrence in local site. So then Chris said, talk to his mentee, Jing He, actually said, why actually let the medical oncology do this analysis? So Jing actually is using the entire cohort to do this analysis, and you can see the curve is totally different. It's not because of Jing He's vision is upside down. It's because of, I'm very optimistic. So I look at how many patients are survived. He look at how many patients will die over each year. So that's why the curve is upside down, and you can see the liver is the first curve, okay? And I guess it's because of this reason, Chris actually didn't pay very much attention, so he started to do the surgery on the liver metastasis. which is called oligometastasis. And so he select a patient who actually are after chemotherapy and remain to be oligometastasis. So he take the patient to the surgery, or he also ask his other colleague to do the surgery. Obviously, after chemotherapy, and the patient had a surgery of this oligometastasis, which is defined by less than 3 or maximum 5 actually metastatic lesion in the liver. And obviously the outcome is better because you're using chemotherapy to select for a patient who had a better biology. But on the right side of this slide, that result is very interesting. So it look at is after the oligoliver metastasis resection, what is the recurrence pattern? Although everybody is recurred, because you can see on the left side, everybody die. But not everybody recur as a liver metastasis, okay? Some patients actually don't have a recurrence in the liver metastasis. They recur in the lung or other organ, and some don't have any organ recurrence. They die. So I think they have other systemic micrometastasis. So that would suggest there's subside of the patient with oligometastasis. They're a new disease. There actually is their metastasis in the liver is a new primary, and then they can actually go to other systemic disease. Same as a lung metastasis. If your first recurrence in the lung, it's a new disease, and then they go to other metastatic site. So that's very interesting, and not surprised. I see the curve in different ways. So I send every of oligo lung metastasis patient to the Steve Young at Hopkins, and he do this resection. Every year he probably can reset at least more than 10 patients sent by me. This is not my study, but I think we'll reproduce the same result. The patient with a lung metastasis after resection have very good outcome. Even without resection, still have a very good outcome. So that suggests the patient who had a lung metastasis, oligo lung metastasis, or has recurrence as a lung metastasis, has a very good biology. And so this is put together, Hopkins data, liver and lung oligometastasis resection, and you can tell actually is a patient with an oligo lung metastasis has much better outcome than the clinical outcome than the liver metastasis resection. So the next question is something genetic played a role, like tumor genetics? One of the T32 trainee mentored by Jing He, okay, Jing actually is already growing to our chief of the HPV surgery. And so he mentored this resident to do this study. I mentor a different resident to do another study, okay? So this study is showing, there is some correlation, but I really cannot find additional correlation of the outcome after the oligo liver or lung metastasis resection. And except that the KRAS G12R, it seems that we are more likely associated with liver metastasis than the lung metastasis. But I do see one interesting correlation is the patient with a liver metastasis, but with a p53 mutation. And those patients, if you take them to the surgery, the outcome is very bad. As you see in the right text, the hazard ratio is 10. So it's almost a crime to take the patient with a p53 mutation to the oligo liver resection. However, small sample size, we have to wait to have more evidence to suggest whether or not we should take a patient with a p53 mutation to oligo liver metastasis resection. So this is telling me, probably not just the tumor intrinsic factor, but maybe the tumor extrinsic factor. So then, again, still Jing and I co-mentor another, resident who came from China. Is mentored by in China by the Professor Yang, and who at the Peking University First Affiliate Hospital, and he's here, I think, in the audience. He did a great job. He's a really very good trained in bioinformatics. So then we take the tissue from this oligometastatic resection and for the combined spatial proteomic, meaning the multiplex immunohistochemistry, and transcriptomic, we're using a non-strain DSP for a tissue microarray. And we have a paired primary tumor and oligometastasis, including paired tumor, oligo-liver metastasis, and a paired tumor of oligo-lung metastasis. On the left side is a representative of CD45 multiplex immunohistochemistry, and the right side is non-strain DSP, and we still incorporate 3 immunostaining on top of whole transcriptome, so we can actually to isolate the cell that is CD45 positive, which is means the immune cell, and or the CD45 negative, which is the epithelium cell. Okay, so first we look at the fibroblast. So this is a very crowded slide. It's very difficult to see actually the result, but in short, liver metastases are less homogeneous with the more ICAF, but and the more ICAF and less MyCAF. So it's a more so that very consistent with what we see in the mouse model, which is I'm going to show you later. While the lung metastases maintain their heterogeneity. Okay. So then the next we see the difference between oligo-lung and oligo-liver is, if you can see, is just the 2 arrows point out. This is the TGF-beta signaling. We look at the TGF-beta ligand, TGF-beta receptor, the intermediate factor like a SMAD3, and the downstream like in the. So this is just one of the results. We're seeing the TGF-beta signaling was decreased in the liver metastasis that associated with the longer survival, which makes sense because it's known the TGF signal is involved in the liver metastasis, but we still had a difficult time to target the liver metastasis with the targeted TGF-beta. On the other hand, the lung opposite, so it's a slightly better trend toward increase of TGF-beta signaling is associated with a better survival. And so the next, we want to see the effector T cell. And this is not a surprise. We're using a multiplex immunohistochemistry to look at a granzyme B positive CD8 T cell on the left side, which is representative of the cytotoxic effector T cell. On the right side, which is using the clustering, just looking at the signature for the effector T cells. And we're seeing the similar result. There's an increase of the effector T cell signal or increase infiltration of the granzyme B positive, the CD T cell in the metastasis, in both the liver metastasis and the lung metastasis, which is not surprise because remember, this is a patient that all have relatively better biology, no matter it's in lung or metastasis or liver metastasis, otherwise they wouldn't go to the surgery, okay? So that's good. That means for that part, they have a very similar biology, but what else? That difference will, telling us that is might explain their different biology. So one is the macrophage. So the M1, M2 ratio, as you know, the M1 is associated with it's anti-tumor macrophage, M2 is pro-cancer macrophage, so the higher M1, M2 ratio should be better. But as you can see in the liver metastasis, this ratio is decreased. So that suggests macrophage is potentially an issue in the liver metastasis. We don't see this in the lung metastasis, okay? So, but what we see in the lung metastasis is the T regular cells. We don't see much difference in the T regular cell in the liver, in the liver metastasis, but we're seeing there is a increase of T regular cell in the liver metastasis compared to the paired primary tumor. However, if there's a decrease of the T regular cell, it's associated with a better outcome. Okay, so that suggests there might be something in the Treg can be a target. But what could be the target? So we look at it through the TGF. Remember, look at the TGF beta, which is associated with the Treg signal, but that's in the liver metastasis, so we don't know. That's not what we're looking at. So then we look at the latent TGF binding protein, which is emerging target for the target TGF beta signaling. So we look at the different latent TGF beta protein here. Just one of that is called a GARP, and then we're seeing GARP positive T regular cell increase in the lung metastasis, and then decrease of the GARP positive T regular cell associated better prognosis. So that suggests it's a potential target for lung metastasis. Okay, so what about the spatial part? And since every nanostring actually can give you some of the spatial information, we did analysis in the multiple small niche in the tumors, and then we cluster them and looking at what cluster associated with a better outcome, what cluster associated with a worse outcome. So the left side is the liver, right side the lung. I have one typo, but it's not important. And so the cluster 5 in the liver metastasis, which is the circled one, the left side, class 5. And you can see the bottom one is showing their correlation with the survival. And you can see this is associated with a very poor survival because essentially every patient who have this cluster don't survive longer, okay? On the right side is the liver. The liver seems to the class 3 associated with the worst survival, okay? And then we'll look at the liver. What is the signaling? And what is my associate with this cluster? What actually is the immune-infiltrated cell? Since we're doing combined nanostring and the multiplex immunohistochemistry, we find actually CD8 cell actually are associated with this cluster. But the CD8 is the effector cell. And then we did a deep diving and find it's not CD8 cell, it's a PD-1 positive CD8 cell. It's actually the CD cell expressed the checkpoint, okay? And that probably suggests, yes, CD8 cell is increased in the metastasis, but they become exhausted when Chris actually resected those patients, okay? And again, what signal? Not surprised for liver associated with a cluster 5 is TGF beta signal. No matter if it's a TGF beta ligand or TGF beta receptor, there's increase in the in the metastasis, okay? And another is, again, if even we use the transcriptomic, we find the same finding. The checkpoint is signaling. For example, PD-1 or and LAG3 or those actually is the immune checkpoint signaling. So that suggests, yes, they have to have a good biology when you do the resection, but the T cell already exhausted in the liver metastasis. That's why they will still recur. And those are all bias analysis. But we also did an unbiased analysis because we want to identify some of the target attributes, through the unbiased analysis, so we can also identify the target. Long story short, and because of the many result is still preliminary, and we then use our the mouse model, which is I briefly mentioned in the beginning. We just did the same thing as we did in the human, and we isolate many cell lines from the mouse KBC model, and find which mice has liver metastasis, which mice has lung metastasis, and we develop the cell line. So we have a pair of cell line, multiple pair of cell line. And here showing if the KPC tumor cell line from the mice with only liver metastasis, if you inject this KPC cell line in the orthotoxic model, even you inject to the liver, yeah, they form in liver metastasis. But if you inject in the lung, they still form the liver metastasis. Versus if the tumor cell lines isolated developed from the KPC mice who has lung metastasis. Even you actually inject them to the liver, they don't form much, very few liver metastasis, they form the lung metastasis. Okay. So then we use this model to test the target we've identified from the unbiased analysis. We found a YAP signaling associated with liver metastasis and those chromatin remodeling signaling, particular PRC2 complex represented by EZH2 associated with a lung metastasis. This is the first mouse study, so it's very preliminary. As you can see, if you're treated, this model is the liver metastasis model. So the tumor cell line, if you're implant into the primary pancreas, they only form a liver metastasis. And then this model actually respond very well to the YAP inhibitor, but not much to the EZH2 inhibitor. Okay. So we're currently actually is doing an experiment on the other model. The model actually is have a preference to form the lung metastasis to see whether actually we see the opposite effect in this experiment. And finally, thank you very much. And in the last few years in Johns Hopkins, actually, after Chris left, Jing and I co-run the lab. So this is all the work we did together, and we continue to collaborate. Okay, thank you very much.
I don't think we're going to aim for questions. Well, thank you, Lei, for that exceptional talk. It actually starts to answer some questions I had about lung-only metastases. I apologize, we're actually not going to have time for the questions, not because of the speakers. We got started late, the video ran a little over, but I do want to thank the four speakers for what were really superb, interesting talks. It's unfortunate that we won't be able to ask questions in an audience, but I'm sure if we track them down, which I'll be doing, they'd be happy to answer any questions. So thank you again, and thanks to my co-moderator here for a great session. Thank you.